Since we've gone through all the alimentary canal and a bunch of the accessory structures, it's time to look more into the physiology of digestion, looking into the four main macromolecules and how they're broken down and absorbed into your system. So first off, chemical digestion occurs in a couple different ways. We have catabolic digestion. In the case of catabolic, looking at breaking down those molecules and releasing energy. Now that can occur in different ways. We have enzymatic. Enzymatic breakdown is utilizing an enzyme, obviously, to break and sever the different bonds. We have hydrolysis. That's the breaking of a bond based on water, hydrolysis, water breaking. And all of this needs to be looked at in the case of different four macromolecules. So we're looking at, first of all, carbohydrates. Now, carbohydrates are going to utilize salivary amylase. Think about the mouth, salivary amylase. Pancreatic amylase. That's part of the pancreatic juice going into the small intestines. As well as various different brush border enzymes. Things like lactase, maltase, there's a whole series of these. But the goal is they're all trying to break down those large carbohydrates into the small individual monosaccharides. Because by breaking them down, those monosaccharides can be utilized to produce ATP inside your body. So your body is trying to take what you ingest and turn it into a usable form. The way you're looking at this carbohydrate transport, because you have to break it down in the intestines, but also absorb it into those simple columnar cells. So we're looking at a secondary active transport. And with what else? Sodium, of course. Sodium is helping transport a lot of these different molecules. So what's going to happen is sodium is going to be moving as well as these monosaccharides at the same time. There's some facilitated diffusion occurring, but mostly it's this co-transport aspect. You're trying to enter in the villi. You're trying to go into the capillary beds. Again, remember we had those circular folds, the villi and the microvilli. They're all about trying to increase surface area to aid in absorption of these molecules. From there, they're heading to the hepatic portal vein. Remember the hepatic portal vein we've mentioned briefly in this chapter, but we mentioned it back with the blood vessels also. The hepatic portal vein is allowing for all of the blood from your small intestine, large intestine, stomach, everything heading from the GI system up into the liver, allowing the liver to process and detoxify. So we're going to have to go through the mucosa, absorption, and head to the liver. So carbohydrate digestion here. What we're looking at, we'll start at the top, looking at ingestion. You're ingesting starches, you're ingesting disaccharides, you're saying here. That's where the salivary amylase starts. So for the carbohydrates, your chemical digestion, as well as mechanical, both initiate in the mouth. Then we also have this pancreatic amylase. The pancreas is going to secrete this into the duodenum, so small intestines. We'll have more enzymatic action at that point. But also, you then have the brush border enzymes in the small intestines. The ultimate goal, go from those more complex carbohydrates down to simpler carbohydrates and eventually into the monosaccharides. You can see how lactose goes down to galactose and glucose. You can see how sucrose goes down to fructose and glucose. And maltose simply goes into 2-glucose. Ultimately, the goal is glucose. Glucose is what's used in your cells to produce ATP. Now, sure, you can use fructose and galactose and these other things, but ultimately, you try to convert them into glucose to be the main energy source going into this whole entire process of ATP production. Next up is protein. Now for proteins, we know we have pepsin found in the stomach. Remember, pepsin was not produced in its active form. It had to be activated once inside the stomach. That means proteins are going to have their chemical digestion initiate in the stomach. Their mechanical digestion, that was back in the mouth when you chew. You then also have the pancreatic proteases. We've already mentioned trypsin, chymotrypsin, and carboxypeptidase. All three of those were starting in their inactive forms. And then remember, once inside the duodenum, they had to be activated, either by the enteropeptidase coming out of the brush border side, or by the activated trypsin itself, or self-activation, or the other two. Brush border enzymes. These are more enzymes that get added into the lumen of the small intestines. So really, again, that small intestine is the main site of chemical digestion for all the different macromolecules. You might start ahead there, but really you're focusing on that small intestine for absorption and getting all those nutrients into your system. Now this is coupled with active transport of sodium again. Again, sodium helps get these macromolecules, these nutrients, into the cells. So here's what we're looking at. We'll start with the individual proteins up top, broken down into the basic building blocks of amino acids. Those amino acids then are trying to go into the cell. They get in the cell by going through this transport mechanism. Sodium is heading in, and it pulls amino acid with it. That's based on the gradient of sodium there. So it's pulling it along for the ride. So it's helping get it into the cell. Now, once you're in the cell, you still have to get into the blood though. So you have to go through the cell, down to the bottom, and this amino acid carrier. It'll take that amino acid out of the cell and put it into the capillary. From there, it's heading through the rest of the bloodstream. So you'll be heading out and up towards the liver. Again, liver's processing here. So what we're seeing in this one, we're starting with our full proteins. We then start with the stomach, that's pepsin. You're breaking those proteins down into polypeptides, you're breaking them down into smaller chains. You're trying to get them as small as possible. But the stomach can only do so much. So you end up going down through the duodenum. Duodenum ends up with the proteases out of the pancreas. Now they're going to take these polypeptides and whatever else is left there and break them down to smaller and smaller pieces, smaller and smaller polypeptides, until ultimately the goal is to make only the individual amino acids. That's by utilization of the brush border enzymes, by the actual enzymes out of the pancreas, and the enzyme out of the stomach. All together, eventually, they break it down to the base building blocks. So a little bit about carbohydrate and protein digestion, sites of where they originate, and then their full breakdown in the intestines.